Hello everyone, my name is Jillian Yamin and I'm a PhD student at the University of Koblenz-Landau and a member of the grad school System Link, which is funded by the German Research Society. And today I'm going to talk about the impacts of invasive plant colonization on riparian soil in southwest Germany. And you can also have, of course, a look on my poster, which will be presented online and at a conference as well. First of all, I'm going to give you a quick overview on the project of System Link. We are looking on anthropogenic influences in riparian areas, especially in aquatic and terrestrial environments. In terrestrial environments, invasive plants are the result of anthropogenic influences. For that, we are looking on two plants. The invasive plant that we're looking at is Sympathins glandulifera, which is also known as Himalayan balm. The native counterpart that we are looking at is Urtica dioica, and they are both differing in their vegetation pattern as well as their root structure. Sympathins glandulifera is an annual plant with a fast vegetation pattern and with a tapered root structure with thick roots. Urtica dioica, on the other hand, is a perennial plant with lateral fine roots that are spreading along the surface. It's also keeping its root structure intact during winter, while the above-ground shoot withdraw during winter. The question behind the vegetation pattern was how the degradation of biomass influences the microbial community and therefore also the carbon pools in soil, such as soil organic matter. In order to do so, we selected 3 transects in southwest Germany, closely located to a river, and we were collecting soil samples by a transect that was either highly populated by Sympathins glandulifera or by Urtica dioica, and we had a third transect that was neither populated by Urtica or by Sympathins glandulifera, which acted as a control in our study. In order to understand the soil organic matter dynamics, we investigated thermogravimetric analysis and differential scanning calorimetry in order to assess the soil organic matter quantity and also the thermal stability. Additionally, we also wanted to understand the microstructural stability, and we also investigated experiments in soil rheology experiments where we assessed the shear stress at the yield point, which provides us information on the structural breakdown of soil aggregation. We ran the analyze for 2 different depths of soil to understand how the root structure actually affects the soil in different depths. On the left side of the slide, you can see the T50 value, which is the temperature where 50% of the soil organic matter is combusted. We were able to report significant differences with the highest values for Urtica dioica, which implies that the soil samples that were populated by Urtica dioica showed the highest thermal recalcitrance. The thermal recalcitrance is also an indicator for the speed of degradation, which means if you have a lower T50 value, you also have a faster degree of degradation, which also results then in lower stability against high temperatures. On the right side of the slide, you can see the shear stress at the yield point expressed in Pascal. And interestingly, we obtained here slightly higher values for Sympathins glandulifera, which is probably linked to the soil organic matter content, which was higher for Sympathins glandulifera. However, I have to emphasize that the results here were not significantly different. Additionally, we also obtained other physico-chemical soil parameters and applied the PCA to understand how the investigated soils behaved and also how soil parameters are related with each other. One relationship that we could identify was the soil organic matter content and the soil structural stability indices, which is already known and reported in literature. Additionally, we also find a negative correlation between the T50 value and the pH. One study suggested that the pH negatively correlates with the activation energy of heterotrophic respiration, which means if you have a more acidic soil, the respiration is inhibited. With our results, we conclude that sampling during vegetation and in winter is also helpful to understand the dynamics of the colonization by Impatiens glandulifera, and also have a closer look on the microbial response by invasive plant colonization.